Thank you, David, for that introduction, and I'd like to start by thanking the organizers for inviting me to this wonderful conference. So here are my disclosures. I'd like to start by just introducing gene therapy in this session, and we can define this for our purposes today as the use of nucleic acids as a pharmaceutical agent. We use things called vectors, which are vehicles used to transfer the genetic material to a cell or host. And typically these are viruses, and today we'll hear about the use of both lentiviruses as well as adeno-associated viruses, which are really the two leading viruses in clinical gene therapy at the moment. Currently, there are two approved and one marketed product. The marketed product is Glybera from Unicure. This is for lipoprotein lipase deficiency, so enzyme involved in fat metabolism. And the other is actually a stem cell gene therapy, and this is Strimvelis from GSK, and it was recently approved in Europe to be given clinically. So this is a very exciting time for gene therapy, but these technologies are really new as pharmaceutical agents, and they're considered disruptive technologies because they obviate the need for the current therapy, which in the case of the hemophilias is protein replacement therapy. And so as of yet, as these products have just gotten approved, there's no real sustainable commercial plan that's been successfully executed, but we expect to see that in the near future. But because this is a new technology and it's still many problems with the manufacturing and implementation, we do expect the prices to remain high. So recently I read a very interesting review written by a former commercial scientist and now entrepreneur and venture capitalist, Jason Gardner. And he applied this thing called the Gartner hype cycle, which is it's a method to evaluate new technologies and sort of look at the path that a technology typically takes. And so if we look at this, we're looking at a plot of visibility of the technology versus time. We see something called the technology trigger. And so this is a potential breakthrough that kicks things off. So early proof of concept stories and media interest trigger significant publicity. But at this time, there's usually no usable products and commercial viability is unproven. But then becomes even more publicity, and then you get this peak of inflated expectations shown here. And this produces a number of success stories, often accompanied by scores of failures. And some companies at this point will take action, and some stay more conservative. And that's also followed by more of these failures, which leads into this trough of disillusionment, where interest wanes and experiments and implementations fail to deliver. And producers of the technology at this point either shake out or fail. But fortunately, often what happens is this thing called the slope of enlightenment that they call, where the technology can benefit from new developments, new modifications, second and third generations appear, and they appear to overcome these early failures, and eventually we reach this plateau of productivity where the visibility is not quite as high as the peak, but substantially higher than where it began. Whoops. And so if we apply this to stem cell gene therapy, it really fits perfectly, where in the mid-1990s, stem cell gene therapy was first applied to T cells for primary immune deficiency. These cells were genetically modified to replace the protein they were deficient in and transplanted into patients. And there was some initial success. And even more importantly, it was shown that this technology could be applied to stem cells, which really opened the door for applying it to other diseases such as hemophilia. And this was really the peak of gene therapy, and not surprisingly, this is when I got into factor VIII research and was quickly drawn towards working on gene therapy applications. But unfortunately, I was not aware of this hype cycle, and quickly it went into this trough of disillusionment where safety concerns came up in gene therapy. As you know, there have been deaths associated with gene therapy, and problems such as insertional mutagenesis were caused by some of these early generation retroviral vectors. And so the technologies really descended back into academic laboratories, such as my own at Emory at this point, and new developments were needed to bring it out of this. And it really came, a lot of this work came out of the group in Milan. Luigi Naldini's group evolved this lentiviral vector technology that didn't seem to have the same safety issues as these early generation gamma retroviruses. And quickly some clinical success stories were brought forth, again in the primary immune deficiencies, but then on to other non-malignant hematologic disorders such as beta thalassemia and now even sickle cell disease. And most recently, as I mentioned, we have now the first approved stem cell gene therapy, Stromvelis. So hopefully now we are in this phase of the plateau of productivity. We're going to see application of this technology to more diseases such as hemophilia. So because gene therapy is now in this phase of productivity, it's become a very crowded space, and this I'm sure doesn't capture all the players that are now involved in gene therapy for either hemophilia A or B. But what we do see is that they're using two main gene therapy platforms. Again, these are adeno-associated virus or lentiviral vectors into stem cells. And this is a collection of both academic groups such as our own as well as several commercial entities. And we'll hear more about the work from Sangamo from Dr. Riley. So I'm going to focus this talk specifically on stem cells, as you're here in lentiviral vectors, and you'll hear more about adeno-associated virus in the subsequent talks. So I'll mention what are stem cells, how are they used in gene therapy, what are the challenges remaining to stem cell gene therapy, what's the status of our program targeting hematopoietic stem cells to treat hemophilia A, and what does the future hold for us. So stem cells can be defined as rare populations of unspecialized cells that possess two defining properties. The first is self-renewal. A stem cell can turn into another stem cell, as well as at the same time have this polar differentiation where it creates a daughter cell which can then turn into other subsequent differentiated cells. And this is the second part of the process, which is this differentiation. Stem cells can become all the cells within a tissue organ or even organism. And so there's really two classes. The first is pluripotent stem cells. These are the stem cells that have the capability to make up an entire organism. Primarily, they come from either embryonic sources, such as embryo blastocyst from the inner cell mast, or now, more recently, they've been shown to be able to be generated from adult cells, such as adult skin fibroblasts. And these cells can be reprogrammed to adopt a stem cell phenotype. But the more common and clinically utilized stem cells are the multipotent stem cells, and the most utilized is the hematopoietic stem cell, and that's where I'll focus my talk today. This cell has the capability to repopulate the entire blood cell compartment, comprising both red cells, white cells, so we have immune function and hemostatic function coming from these cells. So this is a basic schematic of how stem cell transplantation is done. There have been over 1 million stem cell transplants have been done throughout the world over the past 50 or 60 years. And so this has been a very successful technology. Typically it's used to treat cancers or primary bone marrow failure syndromes. And in that case, a donor cell is derived from a healthy individual, and these can just be taken from the blood. And then we actually get rid of the patient's own cells to make room for these cells we're going to transplant. And so we can use radiation, we can use chemotherapeutic drugs to do this. The donor cells are transplanted in, and what we hope to establish is at least a chimeric state where we have a mixture of healthy donor as well as some remaining host cells. So in order to apply this to hemophilia, though, we need to add an additional step, and that is that hematopoietic cells do not produce factor VIII, at least as far as we can tell in my laboratory and several others in the world. So we need to add a step of gene addition in this case, and that's what I'll be talking about today, and we are using lentiviral vectors to do this. So the overall schematic gets a little more complicated when we add in these vectors, but it's basically the same procedure. But now instead of getting donor cells from a healthy individual, we can use the patient's own cells. And these, again, are isolated peripherally, or they can be harvested from the bone marrow. We add lentiviral vector encoding factor VIII to these cells, and this is done in a clinical cell processing laboratory. This is now the drug product. It's actually the cells that are going to be transplanted back into the patient. Again, we need to get rid of some of the cells inside the patient to make room for these new cells, and at this point we again hope to achieve this chimeric state where we have some genetically modified cells that are now producing factor VIII and secreting it into circulation. So there are really four main hurdles that we see to implementation of this therapy. First remains the optimization of transgene expression and product biosynthesis. And as many of you know, if you've tried to make factor VIII commercially or in your laboratory, or you purchase factor VIII and you see the price, you know that it must be very hard to make, and that is true. It's expressed at 100 to 1,000 times fold lower levels than other proteins, such as monoclonal antibodies, which are other proteins used therapeutically. The second thing we need to look at are safe and effective pre-transplantation conditioning regimens. As I mentioned, either radiation or chemotherapeutic drugs can be used to do this. Radiation obviously has a number of side effects, as do chemotherapeutic agents. So identifying transplantation conditioning regimens that have very limited toxicity is one of the main areas of research in this field. The third is clinical vector manufacturing. As I mentioned, these vectors are still very hard to make. We really need some sort of quantum leap in vector manufacturing to make this process scalable at this point. But we are able to do it at sufficient quantity to at least treat patients through clinical trials at this point, and I think at least launch some of these products. But I think advances will be needed to implement these globally. And the last is that we need to ensure safe and efficient gene transfer. So we need to minimize the amount of vector used to genetically modify cells, and also make sure that the stem cells retain their stemness. So first I'll focus on our work on the optimization of transgene expression. And our interest in this area really came out of the pioneering work from Pete Lawler, who was the inventor of recombinant porcine factor VIII, and he was and still is at Emory University. Here's a picture of him as my representation of a human making human factor VIII. But what he discovered is that when you try and make recombinant human factor VIII and recombinant porcine factor VIII using the exact same systems, you get 10 to 100-fold more porcine factor VIII. And so this was the time I joined his laboratory and started to look at this, and what we found is that it's actually a difference in the secretion of the molecule. So, and this is represented in this data shown here, where we're actually labeling factor VIII being made in the cells. We see similar amounts of factor VIII being made inside the cell, but we see much of the human factor VIII is stuck inside the cell and doesn't get out, whereas most of, in this case, our bioengineered factor VIII gets out into the media. So having observed this phenomenon, we were also able to map the sequences that were responsible for this high expression property, and found that they were in two distinct domains of porcine factor VIII. And so by engineering those into human factor VIII, we now have a molecule that we call ET3, which is a bioengineered factor VIII. So as a process of using this in gene therapy, we also generated a lot of material, recombinant material, to study the biochemical properties of it. And in doing so, we realized, quickly that we can make a lot of this protein potentially for very cheap. And so we started a program to actually try and develop a low-cost recombinant factor VIII. And so this is just some overview of data on our manufacturing at pilot scale. And we also looked at the biochemical properties of the molecule, and we see here we get very pure material. Both of these bands are factor VIII. We can activate these with thrombin, so they convert from a pro-cofactor to an active cofactor. It binds VWF, so it should be retained with normal circulating half-life, at least as well as human factor VIII. That's shown here. It has a slightly higher specific activity per milligram protein than human factor VIII. And following thrombin activation, it's actually more stable than human factor VIII. And this is something we had previously observed for porcine factor VIII. It hasn't been shown yet whether or not this will be important hemostatically, but we think there's some clinical evidence based on mutations in human factor VIII that cause hemophilia that suggests that this could be an important property as well. So just to cover this part quickly, we have this protein. It looks and acts like factor VIII, human factor VIII as best we can tell. We can make it in serum-free media, and we can do this very cheaply. So the main concern for developing it as a pharmaceutical would be the risk of immunogenicity from these porcine sequences that we've incorporated, and these are the final studies that we're going through right now. But as initial application, we think that it could be applied for a lower cost immune tolerance induction, and hopefully for globalization of replacement therapy. We've still continued on for this approach to bioengineer factor VIII to make even better molecules, and this study is yet unpublished, but has been accepted for publication, and I expect it to come out soon. And I wanted to bring it up because Dr. Phil Zakis, who's shown here, is a former graduate student of mine who's now working with Dr. Lillicrap, and I expect him to continue to do great things in his laboratory. But in a collaboration with an evolutionary biologist at Georgia Tech, Dr. Gaucher, we've been able to apply statistical models to the existing known sequences of factor VIII and actually reconstruct an ancestral factor VIII tree where we know all these sequences. And because it's now possible to actually synthesize this genetic material de novo, we can actually make these proteins and study them in the laboratory. And so we've done this, and we found that some of these ancestral proteins also have this high expression property, as well as some other properties that will be described in our paper. But we have initially applied these in a gene therapy manner, both using lentiviruses and AAV, and I show one data slide for AAV here. So this is ancestor 53 is a common ancestor or predicted common ancestor of both humans and rodents, and it expresses here at a much higher level than a codon-optimized human factor VIII in this study. So we're not the only ones doing engineering of factor VIII. You'll hear more from Dr. Riley about the work they've done modifying human factor VIII to improve expression, because we all recognize that this is a major barrier. So now we get back to combining this high expression therapy with our stem cell transplantation gene therapy, and we have this schematic here where we're using the lentivirus encoding now our bioengineered factor VIII. We put this into hematopoietic stem and progenitor cells and transplant those and expect a significant expression of factor VIII subsequently. So this provides a potential cure. We know that these stem cells can actually last for the lifetime of an individual based on all the studies that have been done previously, these 1 million plus stem cell transplants that have been done. So we expect this to be a one-time shot. By doing the genetic modification ex vivo, we think we have some control over this process. We're able to look at both the integration copy number as well as potentially map all the integration sites prior to transplanting these cells into the patient. And because we're targeting stem cells, we only need to target a few cells, because each one of these stem cells will give rise to more than 1 million daughter progeny, and each of those cells will contain this genetic material and can now produce factor VIII as well. So we get this great amplification. So I don't have time to go into all the preclinical studies that we've done, but this is really a collection of work been done over the past 12 years or so, and some of the pioneering studies were done by someone in the audience today, Baggy Gangadharan, who I saw prior to speaking, so I wanted to mention him. But he really did the first study that we published in Blood, applying first a porcine factor VIII transgene to murine stem cells and showing that this was efficacious in the Hemophilia A mouse. So now, moving on to more of our preclinical data, we have a lead candidate we call CD68ET3 lentiviral vector, and this is a plot showing some of our final efficacy data. So we've taken now mouse stem cells, we're genetically modifying them with the clinical vector, and we're looking at how much of the transgene does it take to produce therapeutic amounts of factor VIII. And so I've put some lines here to represent the transition from severe to moderate to mild to normal hemophilia. And then I put a bar sort of where the minimum copy number is to start to see therapeutic benefit. And what you see is it's a very low copy number that we need to achieve at 0.02. Now, routinely in clinical lentiviral stem cell gene therapy, we're now achieving levels that are much closer to 1. So we expect this to be therapeutic. So that brings to the next challenge, which is developing safe and effective pre-transplantation conditioning regimens. And we really spent a lot of time looking at this. This is hard to do, because most clinical transplantation is done in a way where a group such as ours at Emory, they used protocols that they've been using for a long time, and they're treating diseases that are not hemophilia. And in the case of a cancer, they may be trying to wipe out all the cancer before they do the transplant. So they may want a more stringent conditioning regimen. So this application of looking for the lowest possible risk conditioning has really not been tested, and it's likely to be done in the gene therapy setting. But in modeling this in the Hemophilia A mouse, what we found is that we could use very low levels of a drug alkylating agent called busulfan, which is used also to treat cancer, but is used primarily as a conditioning agent now in gene therapy applications, and combine that with an immunosuppressive agent, antithymocyte serum. And in humans, there's a similar product called antithymocyte globulin. And in using these two agents, we were able to get high levels of factor VIII activity. We see 3 and 4 units per mil of circulating factor VIII without the development of anti-factor VIII inhibitors. So this is the clinical conditioning regimen that we have proposed to the FDA, and this has been presented in a pre-IND. So the patient is admitted on day -6, comes in, is evaluated on days -5 and 4, and this can be done in an outpatient setting, receives a small dose of busulfan to make some space for the stem cells that we're going to transplant. And then days -3, -2, and -1 is treated with anti-thymocyte globulin to provide some immune suppression to the factor VIII that is going to be generated from these cells. Now, this is something we don't know if this is required or not. It does appear to be required in the hemophilia A mouse model, but unlikely may not be in humans, but as this is a very safe agent, the consensus was best to be safe and include it in this protocol. And then on day 0, the hematopoietic stem cells are infused. Now, during this period, platelet levels may drop slightly from the use of busulfan. Oops. And so recombinant factor VIII will be given to maintain factor VIII levels in the 40 to 70% range until platelets recover if they need to. So looking at our clinical manufacturing, this has been a very interesting journey for us. And I showed this slide. When you are a small academic group trying to do gene therapy, you can't do all the parts yourself, and you need potential partners. And unfortunately, our partner ended up being purchased by a bigger group. We were working with a group called Lentigen, and they were purchased a couple years ago by Melteni, and we were all at the point of having GMP processed vector already made and in the freezer from Lentigen at this point, and we essentially had to start over and find a new partner. This also gave us some time, though, to revisit the manufacturing scheme that we were using, and we actually ended up redesigning our vector to improve titers, because as we scaled into GMP production, our titers dropped significantly, and we knew that we were really at this point where we could probably do a phase 1 trial but not go beyond. But what we found is that by redesigning the vector, we were actually able to increase the titers significantly. And we partnered with a new group at Cincinnati Children's Hospital Medical Center, Dr. Poonam Malik, who was pioneering stem cell transplantation gene therapy for sickle cell disease there. And she had gotten the hospital to buy in and create a facility to manufacture vector. So we partnered with them for this process and now have GMP qualified vector in the freezer. Some of the parameters of this vector are shown here, but the main point is that we think we have enough to treat 11 patients, which we have proposed 7 in our pilot study. So the last point is looking at safe and efficient gene transfer. As I said, we want to minimize the amount of vector required and also make sure that the stem cells maintain their stemness. But another requirement in this field is to actually map the sites where the lentiviral vectors are integrating, and hopefully they're not in regions that are known to be involved in cancer. And so we brought in a bioinformatic specialist into the laboratory. He also knew some molecular biology as well, so he was actually able to put together the entire pipeline from collection of the cells to mapping of these integration sites in human CD34 cells. And some of the integration data is shown here in this Circos diagram, where these are all the human chromosomes arranged in a circle. The blue dots represent sites where our lentivirus has integrated, and the size of the circle represents how many integrants how many sequencing reads we got at that site. And the red serve as internal controls to know that the process worked. And what we saw is that our data was not any different from what was being published clinically from other groups moving this technology forward. So we feel confident in our product, as theirs are showing, will be safe and not have insertional mutagenesis issues. As also one of the preclinical activities we did at what's called a clinical protocol simulation, where we brought in a healthy volunteer. We collected stem cells from this individual, and then in the clinical processing suite, the exact same way we would do it clinically, we genetically modified these cells, but instead of transplanting them back in, we just studied them in the laboratory. And we looked at the copy number that we achieved, and we achieved close to 1 copy per cell in this individual's stem cells. So we think that the transjection process is working very well. Whoops. So that brings us sort of to the end here, where with our proposed pilot clinical trial design, the patient will come in, the cells will be mobilized using GCSF. So this takes the stem cells out of the bone marrow and into the periphery, where they can be harvested from the blood. These cells are then processed, and we select out CD34 cells, which it's a population containing the stem cells. We freeze a backup batch and transduce the remaining cells. The patient is then preconditioned, and transplantation is done. This protocol has been reviewed by the Recombinant DNA Advisory Committee in the United States. It has been reviewed in a pre-IND by the FDA. It has received biosafety approval at Emory, so this will be a single-site trial at Emory University with the potential to expand to other sites, including Cincinnati Children's, hopefully. And we anticipate both IRB and IND submission towards the end of this year. These are some of the players that have been involved, and I would be remiss to not mention the funding from both NIH. Our children's hospital has been very supportive, and Hemophilia of Georgia has provided incredible support for this program. So, as I mentioned, future plans, we're still trying to improve on this as we go forward. We think, this is still a first-generation technology. Clearly, advances can be made, and we're all still working on those at the laboratory. But we do think we have a very strong lead candidate that we can move forward into clinical application. And so that brings me to the end here. So stem cell gene therapy, we think we're in this plateau of productivity where life is going to be good. Hopefully for the rest of my career I can go on and treat many diseases, including hemophilia A, maybe even hemophilia B as well. But now there's some new technologies coming on, and some of you may have heard about CAR T cell therapy for cancer. But what we're going to hear about next from Dr. Porteous is this gene editing. So it will be interesting to see if gene editing can actually ride our plateau, or is this a new peak of inflated expectations? And I'm excited to hear from Dr. Porteous on the advances that they're making. And at that point, I will conclude and thank the many people from the several different groups that have collaborated to put this together. It certainly was not me doing any or all of this. Important members of our team at Emory, including Christine Kempton, who may be here today. I know she's at the conference, who's our hemophilia director, as well as Ned Waller. I don't think I have Shannon Meeks on here, but she's been a great resource for looking at the immune response to Factor VIII as well. And again, funding support from NIH, Children's Healthcare of Atlanta, Hemophilia of Georgia, Bayer, and the Georgia Research Alliance. And at this point, I guess I can take questions.
Sure.
Thanks.